Okay, so the journal this week was about human IPS cell-derived dopaminergic neurons function in a primate Parkinson's disease model. Our figure was figure 2a through c. The primary research question of the paper was if it was possible to use human IPS cell-derived dopaminergic neurons as a treatment for Parkinson's disease in primates, if it's safe and if it's efficient, that sort of thing. Our figure's question was if the transplanted cells would grow enough in the brain to replace the damaged tissues from Parkinson's disease, or if they would, stop growing before they became tumorous in nature. So background, Parkinson's is a neurodegenerative disease that affects the midbrain of humans and primates and such and so forth. And this hits the substantia nigra, which produces dopamine for the body. Without dopamine, signals are transported less, so voluntary muscle control kind of suffers as a result. So IPS cells are cells taken from a human and then turned into induced, pluripotent stem cells, which can in turn turn into basically any other cell in the human body. Prior experiments have been done on rats and other models for this, but they lacked the long-term study, and they were basically just to see if you could transfer the cells in the brain, if they would work. Okay, so dopaminergic nerve cell progenitors are cells that turn into dopaminergic nerve cells. So they're derived from stem cells, and then they in turn turn into nerve cells that were used in the brain. Okay, so workflow. Start off with they got cells from Parkinson's patients and healthy humans. Parkinson's patients, because if they could get these cells from a patient and it worked, then you could just transplant them in. There'd be less chance of rejection because they're basically your cells. So at first they turned these cells into IPS cells, and then those IPS cells into dopaminergic neuron progenitors, and then they sorted them, because not all of the stem cells were turned into dopaminergic neuron progenitors, and even if they did, they had to sort out the ones that had certain markers on them that indicated that they might turn cancerous in the future. Then they took these cells and transplanted them into primate Parkinson's models. They got the models from taking a monkey and giving it neurotoxin in the substantia nigra, which damaged it and its ability to produce dopamine. And then they put the cells in there to see if they would grow back and work in place of the damaged cells. For our figures, they decided to MRI and PET scan them over several times over a period of 2 years, so they could see the growth of it and determine if it was replacing the damaged cells and if it was sort of plateauing in growth, so it didn't turn like cancers where it just keep growing. Next slide, please. This is an example of one of the type of monkeys used in the study. My figure was figure 2a. It's one of the MRIs from one of the monkeys, monkey 11, I think. And this is about a year after the transplant. And the arrows indicate the areas that they grafted the progenitor cells in. So one in each hemisphere of the brain and the substantia nigra. And yeah, they didn't show that much cancerous growth. It looked like they were coming in fine. They were showing other results in other tests. So next slide. This is an example of an MRI. This is a human MRI. I'm not sure if there's a specific one they used for monkeys, or I mean, it could be smaller, but yeah.
For figure 2b, they took the graft size and related it to the time after transplantation. So the graft volume increased as the study went on, and as they approached the 6 to 9 month time period, the graft size started to stop growing and just kind of plateau. So every 3 months, they would get MRI scans and PET scans of the monkeys so they could consistently watch the transplantation growth. And on this side there, you can see that the the vehicle monkeys are the control ones. The healthy monkeys are the monkeys that were grafted with healthy IPS cells, colored blue and purple. And the PD monkeys are the monkeys that were grafted with IPS cells from individuals with Parkinson's disease, colored red, orange, and pink. The monkeys grafted with this, the volume of the transplanted graft was calculated with MRI PET scans along with an equation. In this equation, Y is the transplanted graft size, I is the subject, J is the time, T with subscript J equals time point, K is when the graft reached halfway up to the flattening curve, R with the subscript I and J is the residual, and there are 4 coefficients, beta naught, beta 1, beta 2, and a random effect of alpha with subscript 2i. K is 7 due to the information already provided. And with this information, the scientists were able to estimate the graft size of the monkeys. So my figure is figure 2c. And this graph depicts the graft sizes at the time of euthanasia for each monkey. So if you go back to the previous graph, you'll see that this graph depicts the time of transplantation all the way to the time of euthanasia, and my graph is essentially just including all of the last data points of each individual trend line for the monkeys. So to break down the graph, looking at the x-axis, there is first 2 categories of healthy and PD. And these are just referring to where the neurons were derived from. So the healthy portion, those neurons were derived from individuals that were healthy, and the PD portion, the neurons were derived from individuals with PD. Above that are a row of numbers, and these numbers are representing the individual monkeys, which were all assigned a different number in the beginning of the experiment. So each of these numbers basically is a identifier for the individual. And then above that, the L's and R's just represent the placement of the grafts in the monkeys' brains, which were depicted in figure 2a. And then looking at the y-axis, there's only one variable recorded, and that is the graft size, which in this experiment was recorded in units of volume, in this case in cubic millimeters. And for a little bit of background information, when we're referring to the graft, we are referring to the midbrain dopaminergic neurons that were derived from human stem cells. So that is what is synonymous with the word graft in this graph. Each individual bar represents the final size of the graft in each monkey's brain. So you can see that looking in the category of healthy and looking at monkey number 7's right graft, you'll see that that graft ended up being the largest graft in the entire experiment, whereas in the category of PD, monkey number 6 right graft ended up being the smallest graft. And again, going back to the previous graph, you can actually see that monkey number 7, which had the largest graft, is depicted by this purple starred line at the very top, whereas monkey number 6 is depicted through the yellow line at the bottom. And you can actually see that they initially started off with significantly different graft sizes. So it's not necessarily, there isn't necessarily a correlation between the time of euthanization and the growth rate or size of the grafts, because monkey number 6 was euthanized around 18 months, whereas monkey number 7 was euthanized after the full 24
And yet, even if they were euthanized at the same time, it doesn't look like their trend lines would have ever crossed over. And so it's not a determining factor. Time is not a determining factor of graft size. So all in all, the average value for graft size out of all the monkeys ended up being 39.4 cubic millimeters with a standard deviation of 21.2 cubic millimeters, which indicates that the grafts were unlikely to cause a mass effect, and also that there was a pretty significant variability between each animal as far as graft sizes. So in conclusion, we didn't really find any obvious defects in the methodology of the experiment, and we definitely agree with the way that the control groups were organized and how the data was collected and the results that it provided.